Thanks, Steph. Can you guys hear me okay? Okay, so let's start. So hi, my name is Chinmay, and I'm here with my friends today, Kristian and Johnmark. They're sitting right here. I'm a graduate student at Harvard studying biology, and we're all here today very excited to talk to you about 21st century brain mapping. Okay, so let's, 21st century brain mapping, that sounds like a very complicated title. So let's start at a place with which all of us are very, very familiar. So I need a little bit of help from you guys. So I'm not from Boston, and so I want some help in recognizing who this person is. Okay, so I'm going to show you a picture. Just shout out who this person is. David Ortiz, right? So this person is David Ortiz. He's apparently a really, really famous baseball player. He plays for the Red Sox. That's pretty much where my knowledge of baseball ends. But we are going to see something incredible that he is doing. Okay, so I want you to pay very close attention to what's happening in this clip. Okay, I'm going to show you a clip, and I want you to pay a lot of attention to this. It's really important. Okay, so let's see this one more time because I think it's amazing. And I'm sure a lot of you think that it's amazing as well. So, can someone tell me what happened? What happened in this play? What did he do? He hit a home run, right? So, in the process of David Ortiz hitting a home run, let's think of all that happened. I want you to keep in mind that all these things happened in 3 seconds. It took about a second for the pitcher to throw the ball, and it took about another second for the ball to reach David Ortiz, and then it took him about a second to dispatch it. In fact, it took 5 seconds for the ball to travel from David Ortiz's bat into the crowd. So let's think about all the things that happened in these 3 seconds. So first of all, David Ortiz predicted the path of the ball. So he saw the pitcher throwing, and he predicted what is the way in which the ball is going to travel, where it is going to come, and things like that. Next, he decided how he was going to hit the ball, whether he was going to hit it on this side, on that side, he decided all that. He calculated at what time the ball would reach him. He did not swing his bat a moment earlier or a moment later. It was at the perfect time when the ball reached him. He communicated all this information that he calculated to his muscles, okay? And then finally, his muscles promptly and swiftly executed the commands. Now, while all these amazing things were happening, he continued breathing, he continued digesting his food, he continued standing up straight, which is something that human beings are the only species who can do this. And he continued all his other bodily functions, like his temperature did not shoot up, he was fine. You're still able to do all these amazing things. So I find that this is really amazing that human beings can do this. And you guys might be thinking that, hey, this is David Ortiz, he's super famous, he's super awesome, and that's why he can do all these amazing things in such quick time. But it's not just David Ortiz. All of us do this. So a lot of you to come here would have taken a car, would have driven here to get to Harvard. And you would have encountered this green light, which means that you can go forward and you can drive your car as you're driving it. Then you see a yellow light where you're supposed to brake and stop. Exactly. So having been in Boston, so yeah, exactly. Sometimes around the world, I come from India. People usually start braking and things are it's time to stop. But in Boston, people decide it's time to actually go faster and just go through it. And then you see the red light, at which point you pretty much have to stop because otherwise there'll be terrible accidents. But in any case, think about this. This happens so quickly. There's so many times when people are driving, the light is green, it changes to yellow very quickly, and then it's going to change to red, and people decide what to do in a split second. People from Boston decide to drive faster. People from other places decide to, put on their brakes and stop at the intersection. And again, this happens really, really quickly. We don't spend a lot of time thinking about it, and it happens really precisely. You stop right at the intersection. You don't shoot over and stand in the middle of the intersection or anything like that. And that's really amazing. And let's now try to think of like the central organ which is responsible for all this complex behavior, and that is the human brain. This mushy looking organ is behind all this amazing behavior. It controls and coordinates pretty much everything that we guys do, and human beings can do some pretty amazing things as we've seen. Now we want to understand how the brain is working. We want to understand what regions in the brain are responsible for, David Ortiz being able to do all these amazing things so quickly, for us to, respond to these traffic signals and these lights. And really, to start understanding this, we need to look at what the brain is made up of, because that's the way scientists and engineers figure things out. We break a system up and then we look at its component parts, we study how they sort of work with each other, and then we can explain the behavior. So this mushy organ is made up predominantly of one type of cell. So the cell is called neurons. So the brain is made up of this type of cell called neurons. So some of you might know that all the organs in your body are made up of different types of cells. Like your heart has a special type of cell called cardiomyocytes. Your liver has a different type of cell. In the same way, your brain has this type of cell called the neurons. And as you can see, it looks interesting. So, like pretty much all the cells in the body, it has a cell body and it has a nucleus. But then it has a few really interesting features. So, it has these things called dendrites. So the sort of antenna, the sort of like horns sticking out, the reindeer horns sticking out at one end of the cell. So these are called dendrites. And dendrites are very simple. So dendrites essentially act as the antenna for the neuron. So the way in which the antenna, if you have like cable TV, your antenna is supposed to catch the signal and then transfer it to your TV. In the same way, these dendrites are supposed to receive and catch signals from the rest of the body, the environment, and things like that. Then you have the second, this straight line over here called the axon. So whenever you hear the word axon, I want you to think of a wire. So this is essentially a wire. Your cable is connected through a wire to your TV. In the same way, your dendrite has a wire sticking out, which sort of is responsible for the signal passing. And at the end, you have this thing called the axon terminal, which is the end of the axon, as the name suggests. And this is responsible for throwing the signal out so that the next cell can catch it, or the next neuron can catch it. So there are really three main parts of this: the dendrites, which is the antenna or the receiver; the axon, which is the wire; and the axon terminal, which is the part which sends the signal out in the cell. So, this seems like a very simple setup. And now let's think of how this thing might be able to perform a lot of computation, process a lot of information. So there is one device which pretty much everyone in this room has used, probably even today, which does information processing, which does computation. Can someone throw out the name? Calculator. What else? Computers, exactly. So you use computers to do a lot of information processing today, and every computer, every calculator requires power, requires electricity. In the same way, the brain also uses electricity to transfer information from one part to the other. And this is a little surprising. The first time I read this, I was like, electricity? That does not really sound safe. There's electricity flowing around in my brain, but it's true. Electricity, which is essentially a change in charges, it's a process of negative charge moving or positive charge moving, is essentially what is allowing our brain to talk to different cells and things like that. So now let's see how this really works, okay? So how does the neuron conduct electricity? That's the main question right now. So what happens is that there is some signal. So as you see, the neuron has a lot of negative charge inside, and there's a lot of positive charge outside. Now, whenever it receives a signal or a stimulus from the environment, some of the positive charge leaks in, and then it sort of propagates and travels through the axon, through the wire. Electricity travels through wires, as we are all aware of, and in the same way over here, this positive charge moves through the wire, and then it sort of reaches to the end, and it's thrown out for the next neuron to catch. So it's really simple. It's just a change in the electrical charge inside the cell, which is on this side, and then outside the cell, which is allowing your neurons to conduct electricity and allowing your brain to be powered and send messages to each other. Now, let's go back to our previous example. So we talked a little bit about these lights. Now let's think of the sort of things that happened when we saw the lights and when we decided to react either by stopping or by speeding past it. So the first thing was that you were driving, your eye was able to see that, hey, there's a traffic light there, and hey, look, it changed its color. So first of all, you sensed some information. Second of all, this information was sort of sent to your brain, where your brain decided whether to speed, whether to brake, what to do, and it calculated things and it processed the information. And then thirdly, you actually executed the command. So your brain said, let's speed up. You told your muscles, let's sort of push the accelerator, let's go forward, and let's sort of get out, and whatever happens to the person behind me or the person from the other side, it's Boston, they probably expect it. For these three distinct functions, there are three types of neurons. So the first type of neurons are the sensory neurons. So as the name suggests, they sense different things in the environment. So your sensory neuron senses the fact that the green light is changing to the yellow light or the red light. The second sort of neuron are what are called interneurons. The interneurons actually go and process this information which the sensory neuron sends them. It processes that, computes that, and decides on what to do. But now you've decided that this is what you want to do, you have to go out and execute it. You need to tell your muscles to contract, to relax, to stop, whatever you want your muscles to do. And that's where the third sort of neurons comes into the picture, neurons that are called motor neurons. So they're really responsible for driving, driving the muscle function, and they're connected to a lot of muscles. So let's see a simple example of how these three neurons work in our case. So over here, you have your eye over here. Your eye is able to detect difference in lights using a bunch of sort of receptors or things which are which are behind your eye. This information is communicated through your sensory neurons up into your brain, where the interneurons sort of process the information by talking with each other and talking with the sensory neurons, and then the signal is sort of sent down to your muscles to execute what the brain wants you to do. So it's really very simple. It's sort of a three-way loop. The first way is the sensory neuron, the second are the interneurons, and then you have finally the motor neurons actually executing things. So that's pretty much how our neurons actually talk to each other and help us do really complicated things. Obviously, this is a simple example. For a lot of cases, there are a lot of neurons within your brain and outside in the sensory systems which are sort of interacting with each other. So now let's, we've talked a lot about the brain and how amazing it is, and I thought it might be best to summarize it with three things that I found which sort of really communicate and demonstrate the power of the human brain. So your messages are sent to your brain at more than 150 miles per hour. All these senses that you have is sending information to the brain so quickly. It's 150 miles per hour, 241 kilometers per hour. That's faster than a lot of trains in the US. That's faster than a lot of other things travel. Second of all, once your brain sort of receives all this information, there is like a ton of information which it's getting. The messages that it creates and sends between different parts, between different interneurons and motor neurons and sensory neurons, are more than all the messages generated by all the phones in the entire world. So think of all the iMessages, the WhatsApp messages, the Facebook messages. All the messages are still less than your brain creates and sends across a period of a day. That's how complex and amazing this machine that we have is. And finally, for a lot of complex tasks, your brain is faster and more powerful than a supercomputer. And these are not really you and I don't think of these as complex tasks. So for example, we cannot train robots to open a door. So when you open a door, you sort of have to turn around the corkscrew and then push. That complicated motion, which is in some ways regulated through our brain, it's very hard for a robot to do. We try really hard to program robots to do it, and we fail miserably. And that's why we spend 7 years in our PhD, because, in any case. The next thing is this brain like just made up of all neurons? Is it just a big bag of mush? Or are there separate, distinct parts in the brain? Now, it turns out that since time immemorial, when doctors have sort of looked at human brains, they have cut us up open and they've tried to look at the brain. They've found 3 distinct regions in the brain. This one very big region is called the cortex, which is responsible for a lot of processing of the information. The second one is called the cerebellum, which is shown here in purple. This is responsible for executing, and it coordinates the motor responses that you have. So a lot of those motor neurons sort of are coordinated through your cerebellum. And then the final is the brain stem, or what's called the old brain, which is responsible for pretty much everything else that you do. So it's responsible for controlling your temperature, for making sure that you're breathing right, for all those sorts of things. Your brain stem is very, very important. So now, so far we've talked a lot about the amazing things that the brain can do, but it's really when things go wrong that we truly understand what part is responsible within the brain for a particular function. And sort of the first example of this came back in the 1800s. So for this, I want to tell you the story of this gentleman called Phineas Gage, who we are going to show you a little bit of him later on. And so Phineas, this is 1830s. Phineas is a railroad worker. He's doing his thing, every morning he goes, he lays down the track. He's a nice guy, very simple chap. One sunny day, it was a nice day outside, and he was laying down the track, and he had his iron bar and sort of hitting it and sort of laying down the track. Unfortunately, what happened is that a piece of dynamite exploded. And this dynamite exploded, and this rod, with which he was hitting, just sort of traveled through his face. It passed through his skull, and it landed far away. So it created like a hole through his face, across his skull, and it landed outside, which is sort of been reconstructed here. It's like a gross looking picture, but you can see that the rod traveled this path and then it ended up outside. Now fortunately, they had really good hospitals even back then, and Phineas was taken to one of the best hospitals. He was patched up, and he survived and he lived. He did not die because a rod passed through his brain. The doctors were able to sort of cure him of that. But what happened is that his friends, his relatives started noticing a lot of changes in his personality. He became angrier, he became irritable, he sort of started to become nastier, and his entire personality changed. He became a little depressed sometimes. His personality changed so much that his friends, who used to call him Phineas Gage, started calling him no longer Gage. That was like a drastic shift in his personality. Probably the equivalent of that today would be, tweeting out late in the night or doing other erratic things, all those behaviors. So, Phineas Gage suffered from several changes in his personality on account of his cortex being damaged in an accident. And this was really the first case in which we saw that some damage inside your brain, inside your cortex, can really lead to a lot of changes in your behavior. And we got the first clue that your cortex is responsible for controlling some complex behavior. So let's wrap up and sort of summarize all the things that we've learned. So the first thing is that your brain is an amazing machine capable of computations at really, really quick speed. The second is that there are 3 types of neurons. Your brain is made up of neurons, and that there are mainly 3 types of neurons. Sensory neurons, responsible for sensing the environment. Interneurons, sort of responsible for processing the information. And motor neurons, sort of responsible for executing the functions. And finally, Phineas Gage's story sort of showed us that the 3 main regions in the brain, cortex, cerebellum, and brainstem, seem to control distinct behaviors and functions of the human body. And I think at this stage, I would like to pause and take any questions that you guys might have. Yes?
So before you showed the image of the cell, of the neuron, I was wondering whether like the whole thing was the cell or only like the
Yeah, that's a very good question. The whole thing is a cell. And that's why this is a special cell, that it's not just the front part. The front part is just the dendrite, the end is the axon terminal, but the whole thing is called one neuron. Yeah, sure.
Are you aware of any case studies where damage to the brain caused a positive change in personality?
That's a very interesting question. Sorry?
Can you repeat the
Okay.
question?
So the question is that, do we know of cases in which there is a change in personality, which is a positive change done to the damage, because of damage to the brain? I think this is a great question, and this is probably a great place to transition to Johnmark, because he's going to talk about this exact same question. Okay? So, any other questions? Someone has? Yes?
The Phineas Gage, when they're forward, I went through his brain, did it go through all three regions, the cortex, the cerebellum, and the brainstem? Or did I miss something?
I don't think it went through all the three regions. It went through a particular region of his cortex. And that's the region that it went through. It did not sort of damage the cerebellum or the brainstem.
So is that why he stayed alive, because it didn't finish?
Brainstem, yes, yes. If his brainstem was damaged, you'd probably immediately stop breathing and die. But yeah, it's a great question. Sorry, the question was, did the rod pass through Phineas Gage's cortex or his cerebellum and brainstem also? The answer is that it passed only through his cortex. Yes?
You were talking about the human brain, but
Yes.
in general, the other animals that have brain is the cellular is the same similar, or is
Yes. So the other animals also have brains. Sorry, the question is that we're talking a lot about human beings over here. What about other animals? Do they also have neurons in their brains? The answer is yes. Mice have neurons. Some other animals have neurons. And you'll probably hear more about it in Kristian's talk, in which he's going to explain the different sort of brain like animals that have brains and things like that. So excellent question. Okay, so with this, thank you so much, and I'm going to transition over to Johnmark. Thank you.
So picking up where Chinmay left off, there's a huge amount that we've learned and a huge amount that we're still learning by looking at what happens to people when certain parts of their brain are damaged. But as you might imagine, although not all brain injuries are as dramatic as these, there are some very obvious problems with this approach. First of all, it requires somebody's brain being damaged. Second of all, you don't get to choose where the injury occurs. Nobody got to choose what part of the brain the rod went through. It's purely accidental. So ideally, we'd like to have some method of looking at the brain that's both completely safe and that allows us to look at the entire brain and not just a particular part of the brain. And that's where something called neuroimaging enters the picture, or taking pictures of the brain in action. So I'll fully explain what it is you're looking at as the talk goes on, but just to sort of clue you into what you're looking at for now. In the grayscale, you can see the structure of the brain, and in the red, you can see an overlay showing a measurement of the brain's function, or what the brain is doing. So in this case, the neuroimaging method is something called functional magnetic resonance imaging. And it's not the only way of imaging the brain, but as of right now, it's both the safest and most spatially precise way we have of imaging the active brain in humans. So it's what I'll be focusing on today. So fMRI has been around for about 30 years by now, and that might not sound so cutting edge, but I want to try to sort of impress on you just how exciting this is from a historical standpoint. So humans have always had brains for as long as we've been around, for all these thousands of years. And we've suspected, at least since the ancient Greeks, that this weird squishy organ inside of our heads has some role in giving rise to our thoughts and experiences and behaviors. But it's not until our lifetimes that we've been able to look inside the human brain in action and see how it works. It's sort of like being alive when Galileo invented the telescope. So in my brief time today, I'd like to both talk about what makes images like these possible, how does fMRI work, And second of all, what has fMRI taught us about how the brain works on a sort of fundamental level?
Hey, Johnmark, you don't have to lean that far.
Oh, sorry, yeah, sure.
And you don't have to use the microphone at all.
Okay, sure.
It picks up.
Oh, great.
Yeah.
Okay. So first of all, what do I mean by the brain in action? So just to remind you of what Chinmay said earlier, the brain is ultimately made out of neurons. If you zoom in on the brain, that's what you see. And the main thing neurons do is fire. So what we want then is some kind of picture that tells us how much neurons are firing in different parts of the brain. And if you think about it, that's sort of a hard problem. You can't simply shine a light inside of the brain. Obviously, there's a skull in the way. So we need some kind of a signal that both reflects what neurons in the brain are doing and that can pass through solid objects like the skull. So one useful fact about the brain is that parts of the brain are kind of like muscles. So when parts of the brain become active, they use up oxygen, and as a result, they have to call for more blood in order to refill that oxygen. So just like doing bicep curls at the gym causes your biceps to get a pump, when different parts of your brain become active, they also get a sort of pump. So they call for more blood. So if you can figure out where the blood in the brain is going, you can figure out what parts of the brain are more active. So how do you do that? Okay. So one kind of useful fact about all of this is that when blood drops off its oxygen cargo at a certain part of the brain, that blood becomes more magnetic. And as a consequence, it changes the magnetic properties of that part of the brain. So if you then have a very powerful magnet, you can figure out where in the brain that's happening. So it's important to point out that this is a very indirect method of looking at brain activity. So it's kind of like trying to figure out what people across the United States are doing by looking at this map of the United States at night. So obviously where there's more light, people will be busier. It's a byproduct of people's activity, but you won't be looking at the people themselves. It's very indirect, and fMRI is much the same way. So in this case, this is what our magnet looks like. Some of you might have seen it before. It's an MRI scanner. So fMRI uses the same machine as MRI. The only difference is that it uses different settings so that rather than simply imaging the structure of an organ like the brain, you're also getting a measurement of the brain's function in the way that I described. So in case you haven't seen one before, you can kind of see a bed here. You can kind of see a big tube here. And so when you do an MRI experiment, you lie down on the bed and then you slide into the tube. So you might be wondering where the magnet is. So the magnet is actually inside of that big tube. And it's actually a very powerful magnet. It's about as powerful as the magnets used to lift up cars in junkyards. So as you might imagine, one very important safety precaution for fMRI is just making sure people don't have metal inside their body for very obvious reasons. So with all that in mind, let's look back at this picture I showed you at the beginning because now it should make more sense. So as I said before, the gray shows the structure of the brain, and the red is a measurement of the brain's activity. And now you know exactly what that means. So what the red corresponds to is a magnetic measurement of where the blood is going in different parts of the brain, and that tells us what parts of the brain are more active. So in this particular case, what you're looking at is what parts of the brain are more active when you're trying to maintain information in short-term memory. So if I were to ask you to remember the digits 5, 6, 4, 6, 5, 2, 5 for 30 seconds, these are the parts of the brain that would become relatively more active. So there's one more important question to ask about MRI as a method, and that's what exactly is the spatial scale of MRI? How much detail can it give us? And to sort of explain why this is important, imagine you're shopping for an iPhone. So one question you might want to ask about each iPhone model is how many megapixels the camera has. Because the more megapixels the camera has, the smaller each pixel will be, and the more detail you'll be able to resolve as you zoom in. So for example, if you zoom in on this top high-res picture, you can still make out a lot of detail as you zoom in. Whereas for this bottom picture, it's much lower resolution. So as you zoom in, it just becomes kind of a blurry mess. So with that in mind, just how big are the pixels of fMRI? So in this case, they're actually three-dimensional pixels, and each pixel is about 1 millimeter cubed on each side. Now, that might sound very small, and it is, but just to give you some perspective, each one of those 1 millimeter cubes contains about 50,000 neurons in it. So just to give you an analogy for some sense of scale, if you imagine that the brain is the size of the United States and each person is the size of a neuron, fMRI really gives you information on the level of sort of neighborhoods of neurons. So it can tell you that maybe that Harvard Square is more active, or Central Square is more active, or maybe that there's a parade going through Harvard Square. But it can't tell you about what's happening to any individual person inside one of those neighborhoods. So for the rest of the talk, I'll be sharing a little bit about what MRI has told us about the brain. But before I do that, are there any questions about fMRI sort of as a method? Yes?
People born in the middle of the last century have a lot of metal fillings in their teeth. Will that be affected? Will you zip up to the top of the MRI?
That's a great question. So the question is, if you have metal fillings, will that cause safety hazards inside the magnet? And the question is, and the answer is, it'll never make you stick to the side of the magnet. As for whether it could cause problems, it depends on the particular kind of metal. Sometimes it'll simply cause a distortion in the image, but without causing a safety hazard. Yes?
Does blood get more magnetic when it goes into muscles?
I believe so. So what? The question was, does blood become more magnetic when it goes into muscles? And yes, it does, because it's the fact of the blood dropping off its oxygen that makes it more magnetic, and that happens anywhere where blood goes. So yes. Yes.
How strong is the correlation to be known between the oxygen presence in the blood, in the presence of activity in other chemicals like that in the brain? I'm just thinking it's a magnetic response, and if you got other elements there that also pick up the MRI signal, how well do we know that the oxygen is telling the true story?
Very good question. So the question was, just how good is the correlation between this magnetic signal based on the oxygen and the actual brain activity?
You don't really measure brain activity. You don't have a way to do that yet.
Yeah, very good questions. That's correct. MRI is a very indirect measurement, as I said. So there have been a few different ways of trying to get at just how direct it is. So, for example, people have looked at animals, so they've done both fMRI scans and done more direct measurements of what the neurons are doing. And they found there's a reasonably close correlation. It doesn't reflect every aspect of brain activity, and I don't want to go too far into inside baseball, but perhaps we can talk over the break back there.
So what's magnetic about when scanning the brain and other muscles? Like the magnetic part is the oxygen, it's magnetic charge.
Good question. So the question was, what's magnetic about the blood? Is it the oxygen? So in this case, it's actually the deoxygenated hemoglobin. So you might know that hemoglobin is a molecule in the blood. But the key here is that it changes its magnetic properties when it drops off its oxygen cargo. All right. Moving on to what fMRI has taught us about the brain. So there have been over 40,000 fMRI studies in the last 30 years, and I can't even begin to summarize all of them. But my remaining time today, I'd like to share some studies that I see as particularly interesting or informative. So one thing that made fMRI especially exciting was that it allowed us to ask some very fundamental questions about how the brain works in a fairly easy way. So one very core question about the brain is, are different parts of the brain kind of like silly putty? Does each part of the brain kind of do a bit of everything? Or are different parts of the brain really specialized for very specific functions? And if so, just how specialized are those functions? So you might have heard somewhere that some parts of the brain are involved in seeing, other parts of the brain are involved in hearing. But what you might not know is that it's even more specialized than that. There are certain parts of the brain that are specialized not just for seeing, but for seeing particular kinds of things. And researchers figured this out in a very simple way. So they simply put people inside the scanner, they showed them a lot of pictures, and then they saw if there were any kinds of things that tended to activate some brain areas more than others. And when they did this, they discovered a lot of different brain areas. So they found that there's one brain area that was particularly responsive to faces, another that responded more to places, another to shapes or objects, and still another to words. So different parts of the brain really are very specialized. So one very nice thing about these brain areas is that they're very easy to find in people. They're common to just about everybody. So just to give a kind of a narcissistic demonstration of that, here's a picture of my own brain with my own face area in blue and my own place area in red. Oh, I should say, by the way, the same principle seems to apply for sounds as well. So this is much more recent, but just last year, some people at MIT found that there are some bits of the brain that seem specialized for music sounds, others for speech sounds, and so on. So you might be asking, if different aspects of what you're experiencing correspond to different sorts of brain activity, could we somehow read your mind? And the answer to that is a little bit. So about 9 years ago now, some researchers asked that question, and what they did, so their goal was to see if they could reconstruct what you're seeing purely based on your brain activity. So it's a little bit like trying to reconstruct the movie people are watching in a theater purely by looking at the reactions of people in the audience. So they simply put people into the scanner, showed them movies, and then tried to see if they could reconstruct those movies based on their brain activity. And I'll let you judge for yourself how well they did. So on the left here, you can see what the people in the scanner actually saw. On the right, you can see the experimenter's best reconstruction of that video. So as you can see, it isn't perfect. It's very blurry, as you might expect. MRI only gets you so much detail. But it's often pretty good. You can kind of make out there that it's a face. Can't quite see that as an elephant, but the outlines are kind of in place. Not quite an eagle. The point is, though, it's not bad. That looks pretty good in particular. So in case all this talk of mind reading seems sort of ominous or scary to you, I should probably mention that people have tried to use this technique for things like lie detection and things like that, and it really doesn't work very well. These techniques are very easy to fool. I'd like to go a bit further than that, though. I'd like to talk about how these techniques can be actively helpful for people. So to kind of motivate this, I want to go back to something Chinmay said in his part of the talk. So you might remember that the brain sort of does three things. It takes in sensory information, it processes it, and it sends commands to the muscles to tell them what to do in response to that processing. But let's say that third part of the chain was cut off. Let's say that the brain somehow couldn't communicate with the muscles. So you had somebody who could take in and process information, but couldn't respond to it in any way. So unfortunately, this isn't a hypothetical scenario. There's a very tragic condition called Lockedin Syndrome, where people have damage to a part of their brain that leaves them completely unable to move their muscles, but they remain completely conscious. So the question is, how could you communicate with such people? So some researchers a while ago asked this question, and their key insight was that although these people couldn't control their muscles, they can still control their thoughts. And so they figured out a sort of very rudimentary way of allowing these people to communicate. And they took the following approach. So they slid these people into the scanner, and they gave them the instructions, we're going to give you some yes or no questions. If your answer to the question is yes, imagine playing tennis. If your answer to the question is no, imagine your house. So it went like this, say yes, tennis for yes, imagine house for no. And the researchers knew from past experiments that thinking about doing athletic activities like playing tennis activates one part of the brain, and perceiving or thinking about locations like your house activates another part of the brain. In fact, the same place area I mentioned earlier. And by doing this, they were able to maintain a very simple conversation with these people, sort of a game of 20 questions. Again, this is very limited, and MRI is very expensive, so it's not something you could do all the time, unfortunately. But it gave one sort of channel of communication where these people didn't have any other way of communicating otherwise. So any questions before I move on? Yes?
Is there a reason why, for example, the shape one, you see activity in both the left and the right hemisphere, but for the text is only on the left?
Yeah, great question. So his question was, why are some areas like the shape area on both sides, and why are some areas like the word area only on one side? So those are examples of some real differences between the right and left side of the brain. You might have heard a lot of other sort of differences mentioned in the media, like some people are right-brained and more creative, some people are left-brained and more logical. A lot of that is very exaggerated and not really based on a lot of data. But cases like these are real examples of differences between the two sides of the brain. Anybody else? Yes?
Some people sometimes say that the woman, they can type more vocabulary because they use the two sides of the brain for verbal intelligence or something like that. Is that any difference between woman's brain and men's brain? Now you are talking about different people, creative people or something like that.
Good question. So the question was, are there differences between men's brains and women's brains, particularly with regard to how they use the right versus left side? I don't know anything about right versus left. I think some studies have found some controversial differences between men's and women's brains. It's hard to interpret them or say exactly what they mean. I don't know those papers particularly well, so I couldn't really comment too much on them, but I do encourage you to read more about it. Yes?
So my question kind of leads off of his. Do you know how he said, is a woman's brain different than a man's? How come women are more emotional? Is that part of their brain more functional than men's? Like, not to sound sexist or anything.
It's a good question. So the question was, there are certain differences between men and women. So for example, perhaps women are more emotional. Is that somehow related to brain activity? And my only answer is I don't know. We don't know. It's very controversial. Some people have found some things. Other people have critiqued those studies. It's hard to know what's due to maybe different upbringing versus what's genetic. So maybe we'll know in 30 years and I'll give you a better answer.
So I can follow up very briefly on that. So some studies have shown different regions of the brain differ in size between men and women, but always keep in mind that these studies are only looking at observations. They can never expand the part of the brain and see if someone turns into a woman or a man. We were definitely not there yet. But something really interesting, just a mild tangent to that, is some people have looked at transsexuals, like for example female-female transsexuals, and seen that in some cases maybe female-to-male transsexuals, their brains more resemble males' brains, but as Johnmark has said, a lot of these studies have been refuted, and it's very, very difficult to prove conclusively, as you can imagine. There aren't a lot of cases to look through.
Yep. All right, so far I've been talking about different parts of the brain for seeing or hearing different kinds of things, for sights and sounds, some areas for moving your muscles. But what about higher level ideas? So if you see the word dog or hear the word dog, the image of what a dog looks like might pop into your head. Maybe you'll imagine what a dog or dog sounds like. But there will also be a lot of knowledge about dogs that isn't really sensory in nature that's also activated. So maybe the fact that dogs are furry, that they're mammals, that they're related to wolves, and so on and so forth. So the question is, where is this kind of higher level knowledge stored in the brain? So this is a very complicated question, and I don't pretend that the following study answers it, but I like it because it's very recent and took a very open-ended approach to answering this question. So what the researchers did was, as usual, they slid people into the scanner, and this time they simply had them listen to stories on the radio. And what they were interested in seeing was whether there were any parts of the brain that were activated more by certain words than other words, and whether there were any differences like that that were consistent across different people. And by doing that, they were able to map a kind of dictionary onto the brain. So in this visualization, you can see a bunch of words on the brain where the word corresponds to whether that part of the brain was particularly activated by that word. So you can see how this might be useful. So if a person has to get part of their brain removed via surgery in order to control seizures or something like that, it might be useful to know what sorts of knowledge might be affected by removing that part of the brain. So next I'd like to zoom in on a particular part of this. So if you look very closely, you might have noticed a part of the brain that shows a particular pattern. So in this case, there's this bit of the brain that seemed to have a lot of words like believed or innocent or claimed that seem to have something to do with the thoughts of other people. So it turns out this wasn't a new discovery. This brain area has actually been known about for 15 years. It was discovered at MIT around 2001. But I want to talk about it for a bit because it's a really beautiful example of a brain area that's specialized not for a particular kind of sight or sound, but in this case for a particular kind of thought. In this case, thoughts about the minds of other people. So these are very important kinds of thoughts as we go about our lives. If you're playing chess with somebody, or if you're on a job interview or a first date, or even if you were driving here and had to guess what the cyclist next to you was about to do, it's very important to be guessing what's going on in other people's heads. It's really a core part of how we go about our lives. And it turns out there's a particular brain area that's specialized for just this kind of social thinking. So how did researchers find it? So what they did was they put people in the scanner and they had them read stories and answer questions like the one I'm about to show you. So here's one example. John told Emily that he had a Porsche. Actually, his car was a Ford. Emily doesn't know anything about cars though, so she believed John. And then they would ask the question. When Emily sees John's car, she thinks it is a Porsche or a Ford. And what's the answer? So very simple task to do. But the point of this was to kind of clue people into this mode of thinking about the thoughts of other people. And when they did this, they found that there was a very specific part of the brain that became active. So there's still a lot to learn about this brain area, but there's some very tentative evidence that it might be involved in certain conditions like autism. So the idea is that some of the difficulty that autistic people have in processing the thoughts of other people might be in some way related to differences in how this brain area functions, although that's still a very early area of research. So throughout this part of the talk, I've been leaving something out. And if you think back to what Chinmay said about the structure of neurons, you might be able to guess what that is. So you might recall that neurons have not just dendrites for receiving messages and cell bodies for kind of supporting the cell, but also axons for sending messages to other neurons. And in a way, this is absolutely fundamental. So if a neuron couldn't pass on what it knows to other neurons, that neuron might as well not even exist. So to give you an example of why this might be important, I want you to think about two brain areas I've talked about already today. So first of all, this face area that's active when you see faces, and second of all, this area that's more active when you're thinking about the thoughts of other people. So it might not surprise you to learn that these brain areas are very tightly connected because one very important source of information about the thoughts of others is what's going on in that person's face. So what are they looking at, what emotions are they showing, and so on and so forth. So the takeaway there is that in order to fully understand the brain, we don't just want to understand areas in isolation, but also how those areas interact. In other words, we want something like a highway map of the brain, something that tells us how the brain is, how different brain areas communicate. So in this case, the name we're going to give to this highway is the connectome. So you might have heard of the genome. Just as the genome is the map of the genes in the body, the connectome is a map of the connections in your brain. So what do these connections look like? That wasn't supposed to appear right away. So on the left, we can see just a raw image of the brain. So if you cut into the brain, what do you see? And you see kind of two kinds of tissue. So first of all, you can see what we call gray matter, and that's where the cell bodies are. And you can also see something called white matter. And this is where all the axons are. So it's difficult to see in this image, but everything in white here is really a very tangled mesh of wires. It turns out, though, that you can use different settings on MRI in order to visualize those wires. And when you do that, you get something like this image on the right. So it might kind of look weird at first, like a bowl of Technicolor spaghetti or something, but just to kind of clue you into what you're looking at. So each strand of spaghetti here is really a bundle of thousands of axons that carries messages from one part of the brain to the other. And in this case, the colors are simply a way of indicating what direction these fibers are running. And just to make it a bit clearer, the shape of this I've also painted on the gray matter. So you can see that where the strands of spaghetti end, they're connecting different parts of gray matter. So it's important to point out that this is really a highway map of the brain. It's not a map of every single suburban street. It shows us what are the big connections between brain areas, how neurons communicate, but it doesn't tell us how neurons within a brain area communicate, what you might see as the suburban streets of the brain. It turns out that even at this very macro level, though, there's a lot that this connectome can tell us. So this is a very recent result, but one result from just last year is that your connectome is completely unique, even at this very macro level. It's kind of like your fingerprint. So if you were to scan 500 people's brains and then bring those same people back 5 years later, you'd be able to match up people with their connectomes with nearly 100% accuracy. You might be asking, though, why should I care about this? After all, my fingerprint is completely unique, but my fingerprint doesn't tell me anything about me as a person. In that respect, your connectome is different. So just in the last 2 years, people have found that your connectome can tell you about how flexible of a problem solver you are. So if certain connections are stronger, that predicts you being more flexible at that. How well you pay attention. So if you're very distractible, that may be correlated with certain connections being weaker. And there's even some evidence that there are certain patterns of connections that predict if you're vulnerable to certain diseases. So there are certain patterns of connection that are present both if you have schizophrenia and if you're at risk of developing schizophrenia. So one hope is that you might be able to use methods like this to predict diseases before they happen and thereby treat them more effectively. So just to wrap up, fMRI is a way of safely looking at the brain in action, currently the safest and most spatially precise way in humans. From it, we've learned that the brain is not like silly putty. Different parts of the brain really do seem specialized for particular functions. And finally, by imaging the brain, we can not only learn more about it, but also how to fix it. So that's the end of my part of my talk, but just to kind of give you a teaser for after the break. I mentioned earlier that fMRI can only really give you detail down to the level of neighborhoods of about 50,000 neurons or so. It can never tell you what those individual neurons are doing. In order to look inside those neighborhoods and see what individual neurons are doing, we're going to need a different technique. But to learn about that, I'm going to make you wait for Kristian's part of the talk after the break. So any final questions about this? Or, yeah.
So you talk about the connectome being unique. The person's connectome can change through the years, and like different connections can be strengthened the weekend through that, though, right?
Yes.
So even though each person's is unique, that unique can be unique through the years as well, right?
Correct.
Okay.
Yep. And that's a very interesting area of research, this idea of neuroplasticity, how the brain changes over time, and yeah, that's all in the connections. So, yes.
The connection between connectomes and certain kinds of behaviors. I presume you got that through statistical analysis. Do you have a theory to back that up?
Very good question. So he asked about this correlation between connectomes and behaviors, and he asked if there was a theory behind that. So the way we got these correlations, or the researchers did, was by doing connectomes, and then they would do some behavioral task that measured certain aspects of how a person thinks. For example, how well they pay attention. So I don't think there's a general answer of what the theory is. Every theory is going to be specific to the task. But the idea is if certain brain areas are better at exchanging information, that might facilitate certain tasks over others. Although, of course, it will depend on what particular task you're talking about.
All right, I'm going to ask my question again.
Sure.
Are you aware of any case studies where damage to the brain caused a positive change in personality?
Great question. So the question was, are there any case studies of brain damage causing a positive change in personality? I'd imagine those are much harder to find because in general there's a lot more ways breaking something can make something go wrong than can make it go right. Not off the top of my head, but I'll certainly keep my eyes open for those. There must be one somewhere. Yes?
Actually, I saw a documentary where after a big accident or a lightning bolt, someone's brain changed enormously and made that person into a kind of genius in a particular way, either mathematically or musically.
That's really interesting.
Yeah, it's incredible.
Cool, I'll look that up.
I'll look it up too.
I have a question.
Of course.
I think the dendrites can multiply, but in your lifetime, can you create new brain cells?
Very good question.
I'd like to know because I'm getting older.
So good question. So the question is, can we create new brain cells? Sorry, do you mean can brain cells grow by themselves, or can we as humans technologically make them grow?
Oh no, grow by themselves.
Oh, sure.
Food, exercise.
Yeah, so it turns out that new neurons can be created, but it's a very specific phenomenon. It's only in 2 areas of the brain, in a part of the brain called the hippocampus, involved in memory, and in a part of the brain involved in smell. A bit random, and it's still very controversial what exactly these new neurons are doing. But I think exercise does help. I think that's one. I think they found that in rats or something. Hopefully it holds up in humans too. So yeah, exercise is good.
You can't make any specific recommendations, but
Everything your doctor said is right.
Yeah.
In the middle.
So is there enough data, neural imaging data out there that you can start to see correlations between differences in the way people see, respond to a specific stimulus and psychological personality type testing? So you can, like if somebody thinks of a word innocent or turns on a light switch or whatever it is, different people, different regions light up, and just does that correlate to any kind of other psychological evaluation?
Great question. So the question here was, are there differences in how people's brains respond to certain things, and do those correspond to differences in behavior? So it's a very complicated question, and I'm trying to think of some good examples of that. So, I mean, the connectome data is one such example. As far as activations go, I think people have found that gray matter thickness in certain brain areas can predict certain things. I think certain patterns of activity in the face area I mentioned can sometimes predict how good you are at recognizing faces, for instance. So there are people called super recognizers who can recognize any face. And I think people have found certain differences in those face areas, although I'd have to look up what the exact differences are.
Have there ever been any studies done on how to deactivate certain areas of the brain? Without any physical trauma and other means?
Good question. So the question is if there are any studies about how to deactivate areas of the brain. I don't know about shutting them off entirely. That might be bad. There are ways of doing it temporarily for an experiment. So there's something called transcranial magnetic stimulation, where you can temporarily weaken the activity in a part of the brain. The brain goes back to normal right away. I'm sure through certain drugs you can modulate the activity of certain brain regions, maybe weaken it in some cases. In terms of, are you asking about permanently shutting it off, or just?
I was just saying, does behavior change in the person based on those areas of the brain being deactivated or kind of temporarily disabled?
Yeah, it's actually a great question. I cut out a slide about that very question to save time. I don't know if there's time to summarize it. It's a really great study. Ask me during the break and I'll talk about that. There's a good example.
So I know that the epigenome is unique a lot due to your environmental factors over your lifetime. I was just wondering if the connectome is the same, does it change because of the different environments you have and different experiences?
Good question. So the question was, does your connectome change with experience? And the answer to that is yes, and it's very complicated. I mean, there's no doubt that part of the structure of your connectome arises through your genes. But really the only way the brain can change, the only way you can learn is by altering these connections. So anytime you learn something, it's a result of your connections being changed. It might be on a very micro level. It might not be on a level that you can see with the kind of MRI scan I showed you. But just the very act of attending this talk, for instance, the fact that you remember it in 2 days means that your brain was changed in some way. That has to be the case. So your connectome is really changing all the time. But there are sort of enduring features of it at the large scale that are unique to individuals. Okay, so we're going to break for intermission now. Feel free to talk to me or Chinmay if you have any more questions. Oh, yeah.
Good job.
Thank you.
Good job. And then we took a more targeted approach using fMRI to ask, okay, what neighborhoods or cities of the brain are active during more specific behaviors? So which regions are involved in and responsible for creating behaviors or sensing shapes and faces? Now, however, with both of these issues, we're still missing something. Remember, the brain isn't made up of regions, it's made up of cells. And so we need to get a little bit more detail if we really want to understand what's going on in the brain. And to explain why, we need to, we need to realize that brain regions are diverse. Just like cities are made up of many types of people who have different jobs and different responsibilities, so do brain regions. It's not just a single person who does, a city isn't just a single person who does the same thing, except 50,000 of them. Boston and New York have, they have lawyers, they have bankers, they have scientists, and so do brain regions. Those brain regions contain neurons that each perform very specific functions and only together kind of put together these computations. However, with fMRI, we are kind of ignoring these details, and we're really just averaging the responses between areas. And when we do that, we kind of assume that all 50,000 of these neurons, or every person in the city, is acting together to do this, to do the same thing. And when we do that, we miss all of the other functions of the individuals and the interactions and it's usually those kind of the diversity that comes together to really to create a computation. So with that in mind, our goal then should be not to figure out what the average activity of the crowd is, but to track what the individual is doing and where the individual is going and what their role is, what the individual's role is in some task, as well as how the individual is interacting with all of the other individuals. So to state this, what our goal then is to observe the activity of every individual neuron inside of a brain. And this task, especially if you think about the human brain, it's 100 billion neurons with trillions of connections and interactions between these neurons. This sounds like a task straight out of science fiction. What I'm going to show you is that it's not. We're actually beginning to realize this dream, but we're not realizing it in humans. That right now is still too complicated. If you want to think of a scientist as children learning how to read, then trying to map out the brain of humans is like a child trying to learn how to read by looking at the biggest, most complicated book that it can find, because that's what the human brain is. So if we want to learn how to read, if we're a child learning how to read, we want to find something simpler. And that's what we are doing as scientists. We need a brain that is not the most complicated, largest out there. And so to do that, scientists turn to model organisms. Model organisms are basically organisms that we use to simplify our problems, to reduce the system that we're using into something that really is kind of the bare bones. And the classic model organisms are basically, you have your mice, or usually range from your mice, these zebrafish, to flies and yeast. And usually when you are trying to pick a model organism, you think, what is the simplest organism that I can use to answer my question? If my question is, how do cells divide, then I need something that is a cell and is something that is dividing, which means I can go down, all the way down to yeast. However, so for our question, we need to think about what we need. So we want to map the activity of every neuron in a brain. And hopefully it's a brain kind of like ours. So in that sense, we need something that actually has a brain and has neurons. So yeast don't work for us. And again, we need something that's in a brain like ours. So invertebrates, like flies, they don't really have a brain that's organized with the same structure that we've shown you throughout the talk. So that leaves us with these two kind of classic model organisms left. And of these, the simplest, the one that we choose, is actually the zebrafish, which may seem odd. Maybe not all of you have realized that scientists use zebrafish as a classical tool for understanding the brain. But I'll kind of explain why we do. So it started in the pet store. People looking to use fish for research actually just went to the pet store to use zebrafish, and the reason for they use zebrafish is they're very easy to handle. You can kind of do whatever you want to them and they'll survive. They're very robust. But more powerful than the adult zebrafish, it's actually the larval zebrafish. The reason for that is that it's transparent. So if you want to understand how things like the intestine are developing, the heart develops, you can do that much easier in a baby zebrafish than in a mammal where one, it's developing inside of an adult mammal, and two, you can actually see through the body and actually see all of the cells as they develop. And that includes the brain. The baby zebrafish doesn't have a skull. That means that we can actually use light. Unlike in the human brain where light can't get through that skull, we can use light in the baby zebrafish brain to see it. But this doesn't do us any good, or actually more powerful, or in addition to the transparency, we also have the advantage of its size. The baby zebrafish in length is only a couple millimeters long, and its whole brain fits within a cubic millimeter, which is the size of that fMRI pixel that we saw. So relative to the human brain, it is tiny. You can't even see it if you have it at this scale. And if we kind of zoom out and compare the regions of the brain, we actually see our cortex, or frontal cortex, so we've been looking at fMRI, is a little bit truncated. It's much less well developed in the zebrafish, and that's because it's a simple organism. It doesn't do any of the complicated, it doesn't have any of the complicated emotions or abilities that humans have. But if we kind of ignore that and look at the rest of the brain, we actually see that all of the rest of the fundamental regions are preserved. And this means that and this is because the zebrafish is still able to do the fundamental things that it needs to survive. It is still able to detect that it's hungry. It is still able to see its food, and it's still able to execute the behaviors it needs to grab that food. And so that means that with the zebrafish brain, we were actually able to, in theory, understand a lot of the fundamental circuits that are in place across the animal kingdom to enable these behaviors. So, okay, so let's just look straight into the zebrafish brain and see what's going on. It's still an ugly mesh. It's still 100,000 neurons. You might be able to pick out some cell bodies in here and some nuclei and say, oh, maybe that's an axon, maybe that's a dendrite. But you're going to have a fun time trying to disentangle all of this. And so we need some more tools. And we need tools in particular that somehow generate contrast within this messy mesh of wires. Tools that kind of say, okay, if I have some sort of neuron whose job I kind of know, can I pick those apart, see where they are, and see where they send their projections? Let's say, it'd be like saying, okay, can I see where all the lawyers in Harvard Square, where they are going, where are they sending, traveling to and fro from? So how do we develop such a tool? One of the ways scientists like to do this is actually to kind of look across the kingdoms of life and try to see if there are organisms that have tools that might actually help us do this. And in this case, one of the most powerful tools comes from the jellyfish, which it doesn't have a brain, but it still helps us with brain mapping. And the tool it has, you're not going to be able to see it here, but if you shut off the lights and then shine a blue light, you'll see that it actually, some of these cells, it has cells that shine green. And you might already be thinking, okay, if I can get those neurons to shine green, that might help us. But how is the jellyfish shining green? Some of you may have heard of this thing. It's our friend green fluorescent protein, GFP. And so it's a single gene that gives these jellyfish cells that ability. And so this is basically what it looks like. It's got zoomed many millions of times over. And the way it works is when it's just sitting there, GFP does nothing. But when you hit it with blue light, GFP gets excited and releases green light. So if we can fill up our neurons that we care about with GFP and then get some blue light onto them, then we should be able to visualize them and actually see them set apart from the rest of those messy neurons. So how do we do that? Let's think about again. Where do proteins come from? Proteins come from DNA. So that tells us that, okay, we should think about somehow changing the DNA of the zebrafish to give it instructions to build GFP in these neurons that we care about. And this is the exact approach that scientists have done to do this. We basically take 3 pieces of DNA and put them together. We take one are the instructions for actually building GFP. Then to that we attach an address. And this isn't just like a mailing address that takes it to a specific geographical location. This is an address that specifies which type of neurons we want. The the neurons with a specific job that we're interested in. And then around this, we attach something that if you were here last week, you've already seen. And that's a transposon. This is a piece of DNA that actually allows the rest of it to kind of be cut and paste into other strands of DNA, like the zebrafish genome. So then with our little circular unit of DNA, what we can do is take it and inject it into single cells, where it'll then hop into the genome, and now it's a unit now it's a new message and tool that the zebrafish itself has. And so if we've designed this right, then in our messy mesh of wires, we should be able to spot out these GFP-filled neurons that we're interested in. Let's see what this looks like. We have our zebrafish here. This is the brain with the regions highlighted. We're going to take a look at a cross-section about the size. And we have a specific type of neuron that we're interested in. These are serotonin neurons that some of you may have heard of. They do lots of things. I'm not going to go into more detail than that. And this is what it looks like. So here we've kind of reduced this area to 10 neurons at each side. And you can already see, just with 10 neurons, the kind of crazy mesh that they're forming throughout the brain, where their axons are being sent all over the place. The lights, yeah. I'll shut the lights to generate some contrast. And so we kind of spin it around. We can see where these axons are being projected all across the zebrafish brain. And that's just with 10 neurons. So to summarize so far, we've seen that scientists have this tool, GFP, that is actually allowing us to define those individual roads that we were missing in our connectome of the human brain with fMRI. And so in the zebrafish, as well as some other of the model organisms, we're actually now being able to build up this connectome at a much higher resolution. So, but we're still missing something, and that was what we saw in the first part of Johnmark's talk, is we still don't know what these neurons are actually doing. Why are they, what do they respond to? When are they firing? Why are they firing? So before we get to how we solve that problem, I'll take any questions. Yep.
Do brains become more different when over years? Like, do they over generations?
Over generation. So his question is, do brains become more different over generations? So during a generation, there is a lot of changes. So the baby zebrafish brain is much smaller than the adult zebrafish brain. Across generations, that actually depends a lot on the species you're looking at. So the parents of the zebrafish brain are going to look very similar to the offspring. However, so they'll be almost identical with some changes. If you take a more complicated animal like us, then we'll actually see very different, there will be probably far more changes between the parent and the child. So it kind of depends on what animal you're looking at. Anything else? Yep.
Are there any studies that do comparative analysis between the size of the brain correlating to the intelligence level?
So So he asked whether there are any comparative analyses that look at how the size of the brain relates to the intelligence. So more important than within the same species. So for animals, it becomes very difficult to define intelligence. There aren't any conclusive studies that say in mice where they say mice with bigger brains are more intelligent than other mice.
I have a bit of an answer. So one thing they've looked at is the size of the social group of the animal. So how big of a group they tend to live in. And they found that there's a correlation between how big a group you live in and how big your brain is. So that's led to something called the social brain hypothesis. So the reason humans grew such comparatively big brains, given how big they are, to deal with the demands of more complicated social life. So that's one answer. There's more to it.
Yeah. Yep.
So we discussed this in psychology class, and how part of the brain that once you stop using certain part of the brain, it stops functioning over time. And altering our brain and affecting it in like a bad way? Do you know how like now we use technology so much, so we don't like think as much as we used to, I guess? So like, is there studies that show that we're like altering our brain by like not using certain parts of the brain, so over time they're not going to function the way they do now?
Yeah, so the question here is whether the lack of activity in certain parts of the brain leads to its ability to function declining. And so this kind of gets at the cellular level where connections between neurons do get weaker when they're not coactive, and they strengthen when they are active together, which has kind of been extrapolated now to this idea that when large regions of the brain are not as active, then those connections start to become weaker between neurons. I don't know of any long-term studies looking at the effects of technology. I would defer that to somebody more familiar with human brains like Johnmark. But yeah, it's an interesting question that really does come down to molecular level details. Anything else before I move on? All right. Oh, sorry.
I think there is a rumor or saying that even Einstein only used like 6% of his brain. Is that based on any scientific findings?
That's based off of a doctor in the 1920s who didn't know anything, and it got popular, and movies like it because then it's an easy MacGuffin, but there's no evidence for it actually being true. Yeah. All right, so as I mentioned, we can figure out where these neurons are going between regions of the brain, but what are they actually doing? Our goal is to observe the activity of every individual neuron in the brain. So how do we find this activity? How do we observe the activity of a neuron? One way you might think about going about this is actually still using GFP, but changing it somehow such that its glowiness, its ability to glow when hit with blue light, is affected by whether or not the neuron it's in is firing. And this is exactly what scientists have done. We've engineered GFP to have a switch. And the switch is done by taking our GFP protein and adding a new protein to it, which we'll call CAM. Now CAM is a calcium-dependent triggered switch. When CAM is just kind of here, stuck to GFP, and there's no calcium, which is the same calcium, by the way, that you drink with your milk, then when GFP is hit with blue light, it no longer gets excited, and green light doesn't come out. However, if calcium does come in and it kind of sticks to CAM, then the switch is flipped, such that when blue light comes in and hits GFP, it can get excited and release green light, which is cool. Okay, now we have a way to modulate the ability of GFP to glow. Green shouldn't be a verb, but how does this actually help us observe neural activity? To see how it does, let's go back to our neuron. Here it's labeled with our traditional parts, the dendrite, nucleus, and cell body, and then electricity or electric current is propagated down this axon. And what should be noted is that at rest, when it's not active, the neuron is kind of empty of positive charge, but surrounding it is a lot of positive charge, including calcium. And when the neuron fires, that calcium is actually sucked into the cell, meaning that the neuron, when it's active, is going from a state of having no calcium to having lots of calcium, which is the exact difference that we need to flip our switch. So in principle, what that means is that if we fill up our neurons, all of our neurons, or all of the neurons in the zebrafish, with GCamp, then the glowing will only take place in neurons that are firing. And in principle, we can get this mesh to kind of look like this, where neurons are popping out as they begin to fire. So this would be cool. We get a zebrafish doing lots of, and we can have its brain firing with, but in order to actually learn something from this, we need the firing of these neurons to be tied somehow to some stimulus and behavior. And if you remember from fMRI, this is what they did with their imaging, is they would have an individual would go into the big magnet, and it would be shown movies, and then you could connect that activity to what they were being shown or to whatever task they were being asked to think about or perform. And so this is exactly what I try to do with baby fish. So rather than asking it to interpret some poem about whether or not John has a Porsche, I take advantage of the fact that zebrafish are freshwater fish and they don't like salt. So when I show them salt, they try to wiggle and get away. And so my question is basically, how does the brain kind of pick up that there's salt coming in and turn and communicate to other neurons that communicate to other neurons that eventually tell its muscles to wiggle away. And these fish are kind of stuck to a plate with a little bit of jello, and then I can take them into a microscope where we can shine a blue laser through it and pick up the green light that comes in through these lenses into a camera up here that allows us to record movies of all of the neurons and their activity. So if we kind of take a look at our brain again, you're going to have a side view here and the top view down here. And if we pick a small region, we can zoom in and actually look at, these are individual neurons here. So you have about 30 neurons in this little field of view. Each of these little rings is the cell body, where the dark region is actually the nucleus, which does not contain GCamp. And you can actually pick up each of the individual neurons as they're firing. I don't have this memorized, so I don't know when a firing is about to happen. But there's an active neuron, there's another one. But we're not doing this just for, like this, it looks cool, but it doesn't give us the whole picture. The whole picture is contained in the whole brain. And fortunately, this microscope actually allows us to pick up the activity across the whole brain, and giving us movies like this, where we can actually capture the dynamics of neural activity as it's being hit with salt throughout the entire brain. And you can actually put together what regions are responsible for detecting salt, and eventually, when it behaves, is this a behavior-related neuron? And so if you kind of think about, if you combine the activity profiles that you get with this, with the type specific labeling that we can get with GFP, you can start to actually piece together how the different neurons with different roles are working together to generate this whole brain activity dynamics that you're seeing here. All right. So finally, to summarize my part of the talk, we've seen that GFP can be used to specifically label individual neurons or individual types of neurons, and then we can combine that with GCamp to actually to label the active neurons. And so together we can visualize this with whole brain imaging to start to piece together the way all of this is kind of working together to produce behavior. Now this is all data that's this is something we've only had the ability to do over the last couple of years. So right now we're kind of in this dilemma where we're getting all of this data, but there's a lot of it. We don't know what to do with it. And so it's a major now analytical problem to try to figure out what any of this means. We're now thinking about things on levels of hundreds of thousands of moving parts, and piecing that together is kind of the next frontier of where our brain mapping is going. But finally, where have we been? We've gone from accidents and kind of random lesioning to see that, to kind of try to piece together broad areas and what they might do, to actually finally being able to ask questions with fMRI about what regions of the brain might be responsible for different behaviors and sensing. And now, using model organisms and bioengineering, we're actually finally being able to piece together this puzzle at the cellular level and asking how the whole thing comes together. So, take any more questions?